Welcome to another edition of Ask Excite. I'm Tim Bushnell with Expert Cytometry. Today's question comes from Cliff. And Cliff asks, can you share some tips for your secrets for a successful sort? First one is go talk to the sort team, whoever's going to be running the sorter, to make sure you understand how they're running their system and you can communicate information about your sample as well. You need to know the rough size of the cell, not flattened down under a microscope size, but the spherical size, because that's going to dictate the nozzle size that you're going to be able to use on your flow cytometer. That nozzle size dictates the pressure that the system can run at, and that therefore dictates how fast you can vibrate the cell, the stream, to generate droplets. So that information is really critical. Second, make sure you filter your samples. You do not want to have a clog on the system because that's going to cause delays for everybody and can result in extensive downtime while the clog is cleaned. So make sure you filter it right before you go to the instrument. Make sure you know what type of tube you're collecting into, and if you're going to be doing downstream analysis, you probably want to add take those tubes and coat them with some sort of protein. I typically add my staining buffer and let them sit for at least half an hour before I run on the sorter. And last but not least, be careful if you're using trypsin to take off adherent cells or breaking down tissue. Most people will neutralize trypsin by adding serum, but unfortunately when you add serum back, that tends to give the cells all they need to start clumping again. So you want to avoid using trypsin. I encourage using soybean trypsin inhibitor or purified BSA as your neutralizing agent. You can also add a little bit of EDTA to help eliminate that clumping, take away some of those divalent cations. I also encourage people to add DNase, about 10 units per milliliter. This helps to degrade DNA so that the DNA doesn't cause the cells to clump. But of course you have to balance the EDTA to prevent clumping with the divalent cations and the cell's natural processes, with the fact that DNase does need some magnesium to operate. So those are some of my steps that I always make sure people know about when we're talking about a sort, to make sure that it's successful. Sample prep is so critical. So really focus on getting a good, clean sample preparation. You may want to go through the motions of preparing your sample and bring that sample to the facility to see how well it's going to run on the machine before you actually commit to staining and sorting, especially if you have you're able to get a plentiful sample like PBMCs or something of that nature. So thanks for your question. If you have any more questions, go to getflowtraining.com for more information, or send me an email at askexcite@expertcytometry. Thanks for another Ask Excite, and until next time, stay tuned right here to learn best practices in flow cytometry with me, Tim Bushnell, and Team Excite.